Biotoxins are substances that are both toxic and have biological origin. They can contaminate in food and the environment, which can make harmful impacts for both human and animal lives. This can usually lead to severe illness or even death. Take the marine life as an example. Because there is an increase of seafood in many developing countries, aquaculture tends to increase. Though there is definitely a challenge to facilitate growth and trade while protecting the health of consumers. One of these challenges is that consumers can suffer paralytic shellfish poisoning, which is a type of pyrotoxin that is produced when shellfish eat a large number of algae. Once these shellfish such a high concentration of PSP, it can become dangerous if they are eaten. Then the consumers would suffer paralysis. Worst of all, there is no particular antidote to cure PSP. But do not worry, there is hope. Researchers and biosystem engineers developed methods that could detect the concentration of the toxin. This is simply called the pyrotoxin sensors.
The most crucial topic in regards to biotoxin engineering is seeing if the food is safe to eat. During our presentation, we focus a lot on how toxins affect marine life. The most common problem is PSP. It is imperative that engineers should prevent humans from consuming tainted seafood. One key thing that engineers must do is protect the environment from algae bloom. Many more different diseases come as a result of animals eating a substantial amount of algae. This algae bloom not only harms us by producing shellfish that have PSP, but it's also very damaging to the environment. As you can see in this picture here, where this fish just died. Cell-based biosensors, or CBBS, is the main technology used to detect harmful biotoxins. This type of sensor works by using living cells to help detect toxins in the environment. Both prokaryotic and eukaryotic cells are included in the sensor. The main advantage of CBBS is the ability to see what the exposure to biotoxins and other toxic agents does physically to a cell. Data collected from the sensor is based on toxin-cell interactions. CBBS also offers some challenges in regards to testing biotoxins. One of those challenges is that shelf life of these cells is very limited. The main ethical issue is keeping the disease out. For this, there are many regulations in place, one being the weekly testing of toxins in mollusks, clams, and other shellfish during harvest season. If the result of the toxins is above the normal, the area or farm will be closed off until 2 consecutive results of biotoxins level meet the requirements established by the local legislation.
As for now, not all sensors are made perfectly. The huge leap that researchers and engineers must overcome is to create a suitable detection method that allows an accurate, rapid, and sensitive analysis, which is essential for monitoring the toxins. Most important of all, it should ensure the customer safety. Going back to the example about marine biotoxins, marine algal toxin detection is used to sense a toxin in algae that surrounds shellfish and could become dangerous for humans to eat. But if another molecular component that are not toxic is caught inside of the sensor because of its similar structure, then the number could be an over or underestimate. If the measurement is inaccurate, then there could be a potential harm for consumers.
Another challenge for CBBS is related to serial viability. It is not easy to maintain the viability of sensing cells on CBBS for a long period of time. To address this issue, many different research groups are still working on it. We think this technology is beneficial, but it is not accurate enough because it is difficult to differentiate toxin with other cells that have similar structures. The demand for creating a broad-based sensor is increasing, which is applicable to biosecurity, environment monitoring, or food and agriculture diagnostics. Biotoxin is getting more attention since more and more people are focusing on healthy diet and environmental health. To innovate biotoxin sensors, biochemical and biomedical engineers are still doing a lot of investigation on it.